Thank you for taking the time to listen and watch my presentation. First of all, I'd like to thank the organizer for giving me the opportunity to present at this electronic conference for some of the work that we have done in the lab over the last 10 years. The title of my presentation is ATP Metabolism in Red Blood Cell as Potential Biomarker for Post-Exercise Hypotension and also a therapeutic target for cardiovascular drugs. Besides myself, and Shada is currently a graduate student in the lab, and both Fatima and Brad are undergraduate students studying in Dalhousie and doing some research work in our lab. This is a slide for the graphical abstracts, and it shows the red blood cell concentration of AMP in 3 groups of rats. The first group of rats receive an exercise preconditioning, which is the red signal, and the second group of rats receive no exercise preconditioning, and both groups receive an injection of isoproterenol to induce cardiovascular injury. And the third group of rats is the control, which receive no isoproterenol nor exercise. As you can see from this figure, and the AMP concentration in the red blood cell are much higher in the rats without the exercise preconditioning. With these results, we hope to formulate 2 working hypotheses. One is the breakdown of ATP to AMP in the red blood cell is a potential biomarker for serious cardiovascular toxicity and mortality. And in fact, in those rats which died during the experiment, have much greater AMP concentration in the red blood cell than those which survive from the experiment. And this second working hypothesis is if we can preserve ATP in the red blood cell during a traumatic insult, and that could be a potential therapeutic target for cardiovascular protection. This is a complex but very important slide trying to summarize ATP and adenosine metabolism in the body. What is believed in the event of ischemia, intracellular ATP is rapidly broken down to ADP, AMP, and then to adenosine by endonucleotidase. And adenosine will then be released rapidly from inside the cell into extracellular space. Another source of extracellular adenosine can come from a direct release of ATP from inside the cell to outside the cell, and then it will be metabolized to adenosine by ectonucleotidase. Adenosine outside the cell will carry out its cardiovascular function, so once it's completed, it is rapidly taken back up inside the cell by nucleoside transporters. And then once inside the cell, it is metabolized to other oxypurin metabolites such as inosine, hypoxanthine, and uric acid. It's important to note that there are these reversible transporters for each one of these oxypurin metabolites. So what we find, hypoxanthine uric acid in plasma, could in fact be coming from intracellular source. Adenosine inside the cell could be used to recycle back to ATP and by this salvage pathway. And this is a very rapid and efficient way of regenerating ATP, as it does not need to go through making ATP from scratch as involving the glucose metabolism, say for example. So knowing the importance of adenosine and ATP in cardiovascular homeostasis, any strategies that would alter this metabolism could have an impact on cardiovascular homeostasis and can be exploited as the therapeutic strategies for cardiovascular protection, and that is the thesis of our current research direction. This slide trying to show the results of exercise on red blood cell concentration of ATP and also the effect on blood pressure. Keeping in mind the treadmill exercise for 15 minutes at a speed of 10 meters per minute and 5% grade is a relatively modest level of exercise for rats, and in fact after the exercise the rat can still jump around and not feeling any tired at all. Even with this very modest level of exercise, you can see the response to exercise in terms of increase of ATP concentration is almost instantaneous. And there's also a very good correlations between the red blood cell concentration of ATP and also GTP, and the correlation is value is close to 1, and suggesting that the ATP concentration could be directly related to lowering of the blood pressure. This slide shows the effect of exercise on ATP concentration in red blood cell and also on the changes in blood pressure between the normotensive sprague-Doherty rat and also the spontaneous hypertensive rats. As you can see from the left side figure, exercise causes an immediate fall or dip in ATP concentration in the red blood cell, and the dip is much greater in the spontaneous hypertensive rats compared to the SDR. But after exercise, the ATP concentration rises. The increase in ATP concentration appears to be much faster in the SHR compared to the SDR. So in other words, a bigger dip of ATP concentration during exercise, but also greater increase of ATP concentration after exercise. If we take a look at the diastolic blood pressure changes, and during exercise there's an increase in blood pressure for both SHR and also normotensive SDR, but the increase is greater in the SHR. And following the exercise, the blood pressure would fall, and the fall in blood pressure is also much greater in the SHR compared to the SDR, suggesting that the post-exercise hypotension or post-exercise effect is much greater in the spontaneous hypertensive rat. The hypothesis that the spontaneous hypertensive rat has greater post-exercise effect compared to the normotensive rat is further supported by this slide. In this slide, I try to draw a correlation between the post-exercise blood pressure and also red blood cell ATP concentrations. And even though both SHR and SDR shows good correlations with correlation coefficient close to 0.9, and the slope of that relationship was much steeper in the SHR compared to the SDR, suggesting that that post-exercise effect greater in the SHR could be attributed to a greater increase of ATP concentration in the red blood cell. Interestingly, the concentration of ATP in the red blood cell we find in rats are comparable to those found in humans, as reported by Dusinska and her colleagues. And the ATP concentration in human is roughly between 1 to 5 millimolar, and the ADP concentration is roughly about 10 to 20% of the ATP concentration, and AMP concentration is significantly less compared to ADP or AMP concentrations. And the other interesting part in this slide is that during exercise there is an increase in production of ATP. And if we take a look at the ATP to ADP ratio, immediately after exercise the ratio is already higher than before exercise. and the ratio is even much greater shortly after exercise. And so is the ratio of ATP to AMP, suggesting that exercise indeed increased the production or metabolism of ATP in the red blood cell in human. Since now we have shown exercise increase ATP concentration, so we want to follow up and to find out if the increase in ATP concentration in the red blood cell from exercise would have any cardiovascular protective effects. To study that concept, we establish a acute myocardial infarction model. In this model, we inject isoproterenol subcutaneously, and then we collect a series of blood samples to measure ATP metabolism in red blood cell. And in this model, because of so many blood samples taken, the mortality rate is close to 50%. In this model, it's important to point out the rat need to be freely moving, and you need to have access to free drinking water. Otherwise, all the rat would probably die. If we take a look at the two figures, looking at the hemodynamic effects after isoproterenol, and there's an increase in heart rate instantaneously after the injection, and the blood pressure would dip dramatically right after isoproterenol injection, but it will rebound back after 1 to 2 hours, and then it will fall continuously during the experiment. Compare the rats that actually survived on the experiment and those who could not survive. There's no difference in the heart rate increase in those two types of rats. However, if we take a look at the blood pressure rebounds, those rats that actually died had much greater or faster rebound in blood pressure compared to those who survived from the experiment. This slide shows the effect of isoproterenol injection on AMP concentration in the red blood cell and also ATP concentration in the red blood cell. As you can see, in the rats which could not survive, or the so-called victim rats, had much greater AMP concentration than those who survive, and they also have greater ATP concentration than the survivor, and suggesting that both ATP breakdown in the red blood cell and also a faster rebound of blood pressure after isoproterenol could be biomarkers, surrogate biomarkers for serious cardiovascular toxicities or even mortality. This slide shows, in addition to the red blood cell concentration of ATP and the other nucleotides, it also shows plasma concentration of adenosine, hypoxanthine, and uric acid. After isoproterenol injection, there was an increase in both adenosine concentration and also uric acid, which could be attributed to increase in breakdown of ATP to AMP. But we don't know at the moment why there was a decrease in hypoxanthine concentration during cardiovascular injury, that is after isoproterenol. It is possible that hypoxanthine might have a specific physiological role to play during cardiovascular injury, but that is just our speculation at the moment. This is a concentration time plot for the nucleotide concentration in the red blood cell and also plasma concentration of adenosine and the oxypurin metabolites. As we have shown earlier that the concentration of ATP after isoproterenol is lower, but the ATP and AMP concentration in the red blood cells are much higher after isoproterenol. Similarly, plasma adenosine concentration and uric acid are much higher after isoproterenol. But interestingly, if we take a look at the time to maximum, or the so-called Tmax of adenosine and uric acid in plasma, they are much shorter compared to the Tmax of AMP or ADP, suggesting that the plasma concentration of adenosine and uric acid are perhaps coming from not only the red blood cell, but also from other tissues as well. In order to study the effect of exercise for cardiovascular protection, we need to create a more rigorous exercise condition, such that by the end of exercise, the rat will become very tired. We find if we exercise rat under this condition for 15 minutes at this incline, the rat will become very tired at the end of the exercise, as they would just stay on the treadmill and not moving at all. It has been suggested that free exercise intensity is more important than exercise frequency for cardiovascular protection. So we create two exercise conditions for different intensity. One we call low intensity exercise, which is for 15 minutes at 10 meters per minute with a 10% incline. And the other intensity condition is what we call a rigorous exercise, which is the condition that we showed you earlier in the previous slide. And under those two exercise conditions, there was a reduction in mortality from 5 out of 10 mortality to 2 of 7 or 2 of 8 with the exercise condition. 1 hour after the exercise, there was no difference. Look at the hemodynamic parameters between those with exercise and those without exercise. However, if you take a look at the red blood cell ATP concentration, red blood cell ATP concentration was much greater in the low exercise intensity group compared to the other groups, suggesting that ATP metabolism is affected by the intensity of the exercise, not the hemodynamic effects. Remember, we hypothesized earlier that the rebound in blood pressures and also breakdown of ATP in the red blood cells are both potential surrogate markers for serious cardiovascular toxicities. And this slide shows the effect of exercise preconditioning on cardiovascular protection. And those with an exercise preconditioning, and there is a much gradual rebound in blood pressure compared to those rats without an exercise conditioning. If you take a look at the AMP concentration in the red blood cell, again we show with exercise preconditioning, and there is a much less production of AMP compared to the rats without the exercise preconditioning, supporting the idea that both rebounds in blood pressure and also breakdown of ATP in the red blood cells are potential surrogate markers for serious cardiovascular events. We also want to show you the effect of a calcium channel blockers, diltiazems, on cardiovascular protections. And to do that study, we use a freely moving rat model of myocardial infarction. As you can see, with diltiazem given by 10 mg per kilogram as a subcutaneous injection for 5 doses, and it reduced the production of AMP compared to those without diltiazems. If we take a look at the rebound in blood pressure, again it shows a reduction in the rebounds in blood pressure after diltiazems. But there's not much difference in terms of the effects on the heart rate after diltiazem, suggesting that diltiazem will reduce the breakdown of ATP in the red blood cell and also reduce the rebound in blood pressure after isoproterenol. The mortality rate in the control group was 50%, and diltiazem also reduce the mortality to less than 20%, even though the size of the study group is relatively small. I'm hoping to make 4 points in this conclusion slide. The first point is ATP metabolism in the red blood cell is a potential biomarker for post-exercise hypotension. The second point is the breakdown of ATP in the red blood cell is a potential biomarker for serious cardiovascular toxicity and even mortality. The rebound of blood pressure induced by isoproterenol is a potential biomarker for serious cardiovascular toxicity, so similar to the breakdown of ATP in the red blood cell. The fourth point is preserving ATP in the red blood cell in a traumatic situation is a potential drug target for cardiovascular protection. There are some challenges and opportunity in this area of research. The challenges will include instability of ATP and adenosine in the blood samples. So because of the instability, we have to collect the blood sample very carefully to avoid damage to the blood cell. And once these blood samples are collected, and they will need to process immediately after collection to avoid in vitro breakdown of ATP and adenosine in the blood samples. The opportunities are quite huge in this area of research, I believe. We could apply this concept in disease and health management and utilizing that ATP metabolism in the red blood cell could be a measure of inner energy, the reserves, and also for cardiovascular homeostasis. It could use in the management of many cardiovascular and metabolic disease, and that could include cancer, aging, strokes, and other neurodegenerative diseases. It could also be exploited in drug development, as we have shown for the antihypertensive drug diltiazems. It could also be used for other cardiovascular agents that include things like the angiotensin receptor blockers, and antiplatelet agents, anticoagulants, and perhaps many others as well. It could also be used to study the cardiovascular toxicities of anti-cancer drugs and also study the value-added potential for many antibiotics and anti-inflammatory agents for the effects on energy metabolism. And finally, we could also use this concept to enhance the safety and efficacies of complementary medicines, and that include the natural health products, traditional Chinese medicine, as well as many energy supplements are currently on the market. This is the final acknowledgement slide. Like every research, it needs staffs, graduate students, and undergraduate students to carry out the research. In addition, we also need collaborators, both from within Canada and also outside Canada. And research will also need financial support from funding agencies in Canada and also from commercial companies and who have donated and provided support to our research over the last 10 years. And I'd like to take this opportunity to thank you for your attention to this presentation.